excited on behalf of the MAPS committee that you're here. It's impressive to see so many of you on a Friday night. I know that extra credit really helps. So welcome. We hope that you enjoy our talk tonight. I have the honor of introducing our speaker for the night. Dr. Douglas Weiser is a professor at University of the Pacific in Stockton. So he is serving as the co-chair of the biology department. He's also director of their graduate program in biology. And I had the privilege of studying and researching under him for about 3 years during my time in graduate school while I was there. So he teaches all kinds of things from pharmacology to general biology, genetics, biochem, it goes on. And so we're excited to have him give us a presentation on his research tonight. So if you could welcome him.
All right. Cool. Thanks, everybody, for coming. It sounds like you can probably hear me. I can sort of hear myself. So, so I'm just going to talk today a little bit about my project. So I'm really interested in cell signaling, so how cells communicate and how they receive information. And I love to study pathways that often don't make any sense, and that's why I'm studying them. So this it has a bad reputation for not being the simplest of topics, but I'm trying to study a pathway that can respond to different things. So basically, this is a pathway that can help a cell respond to stress. A cell is experiencing something that's not favorable to it, and it can try to repair that damage. Okay? But at the same time, the same pathway can actually signal that maybe that cell should just die, and it'll actually signal to apoptosis. And the same pathway can really help make those decisions. I'm going to talk a little bit about the kind of experiments that I've done over the years, just a little bit about a few things that go into that area. Okay, and I just want to just give a little background on myself. Is I always knew I wanted to do science. I've liked a lot of different aspects of science, but I was never sure what I wanted to do. And I was one of the weird people that really liked chemistry in school, and I was a chem major actually, and I loved it. But my hobbies were often biology. I was the kind of guy who likes to catch snakes and frogs on the weekends, and still do that. But when I was in college, for me, one of the things that really had a big impact for me was my older brother got cancer when I was in college, and that had a big impact on me about what I wanted to do with my career, and I focused on cell signaling and kind of combined my interest in chemistry and biology and went in and got a PhD in pharmacology and cancer biology, and that really was my focus, and I've kind of continued that up until today. Okay, and so what I want to talk about is try to start with some basic biology ideas that you might be familiar with from essentially a textbook this was working. Sorry. When I was testing it, it was working. Okay, so I just want to talk about homeostasis. So this is literally a textbook picture that I put up on homeostasis, right? And this is an example that's not something I'm going to talk a lot about when it comes to insulin and glucagon, but it's a really good example of everyday homeostasis, of why the body's trying to maintain a particular balance. So we have a certain amount of blood glucose that we want to have, the amount of sugar in our blood that's necessary to keep our brain happy and keep our basic biology working. Okay? We don't want it to be too high, and we definitely don't want it to be too low. Okay? And of course, it's actually pretty hard to just eat exactly the right amount of glucose, just have a drop of glucose every 10 minutes just to keep your blood glucose perfect, right? That's going to be a challenging thing to do. So often when you eat a meal, you generally are going to eat too much at once. So that's going to kind of throw you out of homeostasis. You're going to have too much blood glucose. So of course, one of the natural things is going to be our body will release the hormone insulin. And insulin will signal to multiple tissues in your body, especially muscles and liver, and they're going to suck up glucose out of your blood and store it as glycogen. And then that's going to drop that blood glucose back to the normal level. Okay? But of course, you can have the opposite problem for homeostasis, which is maybe you went to bed for the evening and you don't eat anything all night. You wake up in the morning, you skip breakfast. And now you've gone a long time period without eating, and your blood glucose is pretty low. Okay? And that's also quite bad for your body. And so we will do essentially the opposite and release a hormone that does the opposite function. In this case, glucagon is going to signal especially to your liver and encourage it to release glucose and raise that up in your blood. And that kind of helps maintain that kind of basic level that we're doing. And that's a really good example of homeostasis, which is it doesn't just happen, right? We have certain levels of any process in our body has to be actively regulated. We have to have signals to tell it to do what it does, input energy, eat meals, do things to keep it at the level where it needs to be. And things can go wrong with these things. So I think we all know diabetes, for example, is one of the most common diseases in the world. And it comes from, there's various types of diabetes, but they often involve something when it comes to insulin not working properly. So it can be fairly mild up to very severe. You can have it where insulin isn't working quite as well as it should be, all the way up to, for example, type 1 diabetes, where it doesn't exist and doesn't work at all. Okay? And so what we can do in a clinical setting is we can tweak these systems. Okay? And I like to refer to these kind of systems as feedback inhibition, where you basically have too much glucose, so you send information to send that glucose back. Okay? So it's a feedback to come back and send information back to the system to get there. And so, for example, when it comes to type 2 diabetes, we routinely have treatments that make insulin work a little bit better. So we're out of homeostasis a little bit, things aren't quite right, your blood glucose is a little too high. We can use drugs to make it work a little better, make that feedback loop work a little better, and get our blood glucose into the normal levels. Okay? But of course, you can also have problems where your blood glucose gets too low. Right? If you, it's very common with people with diabetes, you might accidentally give yourself a little bit too much insulin, things like that, a little bit too much treatment. You can try to fix that by either consuming a little bit of extra sugar, eating a few pieces of candy or fruit, or in an emergency, you can actually inject glucagon itself and boost your glucose. Okay? And so that's an idea of one context of homeostasis that most people see in like a classroom kind of setting or know people with diabetes. But it's important to note that homeostasis also comes in at a lot of other places. So I kind of want to talk a little bit about cellular homeostasis and as far as cells make decisions. And this is another kind of textbook decision. And we might imagine that if a cell, especially something like a bacterial cell, if I just had a flask of really good nutrients for bacteria and I let one little bacterium in there and let it divide, one cell would divide into 2, and then the 2 would divide into 4, and then into 8. And very rapidly, you just have a massive number of cells in that culture, and they would go crazy. And bacteria do that. And that does happen in biology, especially like diseases spreading and things of that nature. But in our bodies, as animals, we definitely want to avoid that. We don't want our cells to grow uncontrollably. We don't want cells to just divide whenever they have enough glucose. Because I kind of just mentioned in the last slide, we try to keep enough glucose around in our blood. We're always the same temperature, we're warm-blooded. We always are breathing oxygen. We don't want that to be the thing that tells cells just to divide. So what we often do is we do a number of things to keep this from happening. And it becomes essentially an idea of tissue homeostasis, where we have a bunch of cells in a tissue, and we want to make sure that we have, again, kind of a feedback loop, and we have a choice between different possibilities. And in this case, we might have a possibility of cell division, but often there's a strong crosstalk between cell division and cell death. Okay? And the cell death is just kind of marked here with just a little hash mark across that cell. And that's something, unless you study cell biology a lot, it doesn't necessarily seem logical. We often think of death as just stuff goes wrong. But in life, in a multicellular animal, programmed cell death is a very common genetic program. We deliberately tell cells to die all the time. Our immune system, our nervous system, all these things can't function without a lot of cells literally dying when they're told to, as they're forming a lot of these connections. Okay? And so what that ends up meaning, because you often have a strong link between cell division and cell death, we often don't have cells dividing in an adult organism unless another cell dies. And we typically kind of replace that. We keep things under control. Okay? And so as far as another disease that comes up that could be a really big problem with tissue homeostasis is when that balance gets out of whack. Okay? So diabetes is a disease of cell communication errors. Well, so is cancer. Okay? And so in the case of this kind of cartoon, we might have a very normal tissue that's kind of dividing. Some of the cells are dying, some of the cells are growing, and we have this sort of normal system that our tissue is behaving. But in a subset of those cells, something goes wrong with the genetics. They pick up certain mutations, usually several mutations, that are going to happen to that cell. They're going to break those proper signaling controls of behavior. And usually what will happen is they will then begin to proliferate too much. Okay? Actually, luckily for us, what will happen is they will still die. That cell death mechanism still exists in cancer cells. It doesn't mean they'll just grow completely exponentially. They grow quite a bit slower than that, luckily. But there is still higher levels of that. And it starts usually with just a single cell that picks up that initial mutation, and then you have all these daughter cells that then begin to come from that grow into the eventual tumor. Okay? And cancer is such a common disease in multicellular animals. It's a major cause of death in the world for humans, but it happens to animals too. And throughout evolution, we've actually had to take this into consideration very heavily when it comes to how cells deal with things. And one thing that living animals have done is they have the ability to sense that something is just kind of wrong with this cell, right? This cell, there's just something that isn't right, right? This cell has some of its signaling is just out of whack. It's too far away from homeostasis. And in an adult human, we have well over 1 trillion cells in our body. And if you're given the choice for the survival of an individual, would you rather have that one cell just die and get rid of it and live with the rest? Or would you rather give it a chance to develop into a cancer? And what you want to do is pretty much kill it. And so we have a very strong sense that when things are wrong, we like to kill those cells off. And a programmed cell death that comes in from a stress-induced, we kind of sense that, and I usually refer to stress in a cellular context as any time we're out of homeostasis. Something is just really messing with things, we detect that, and if we can't fix it, we typically kill the cell. Okay? And that plays a really important role, and it dramatically reduces the amount of cancer that a person will get in their lifetime. It obviously doesn't eliminate it, because it's still a disease that happens, but we evolutionarily have put a lot of brakes on the probability of that. And it's one of those things that's kind of crazy. If you take away one of the brakes that I'm going to talk about next, when it comes to cancer, a good percentage of humans that are born with mutations in this actually can develop cancer before they're even born, right? And so we actually have very strong mechanisms to prevent cancer, and a lot of the cells that develop into that die before you get there. Okay? So this is a really important molecule in cell signaling. It's got a kind of boring name though. It's just called P53, okay? And that's just the molecular weight the protein originally had. But it's a super important controller of stress signaling. Okay? It plays this very central role, and it really is good at detecting that something is wrong with that cell. Okay? And so one of the really famous things that it can do is it can detect that there's something wrong with the DNA. Right? The DNA has damage in it. So for example, if you're exposed to ultraviolet radiation or ionizing radiation, so this would be, for example, X-rays or gamma rays versus UV just going out in the sun, those will cause different kinds of DNA damage. Those will typically activate this gene called P53. Okay? And P53 as a protein will signal and do multiple different things to the cell. And it's a really good example of this response to stress that I'm very interested in. And one of the very first things it does, and very strongly, is activate DNA repair, right? Which goes back to what I was kind of talking about. Something is wrong with the cell, we've got damaged DNA. Things are bad. That's a pretty high risk factor that we might have accidentally mutated our DNA in a way that might develop cancer. So what we should probably do is just pause for a minute and try to fix that, right? Let's just fix it. I mean, if we can fix something that might give cancer, why not, right? So we'll pause and do that. However, not every DNA damage can be repaired. Sometimes there's too much of it. Sometimes you really have developed already toward cancer. And we know that that cell is developed toward cancer, and we actually call that oncogenic signaling, which is where we know we've made a step toward cancer, right? And we have damaged DNA, we're making mutations toward cancer, that cell's getting, honestly, too much signal to divide. You know what? For the good of the organism, it's probably better to signal to apoptosis, which is programmed cell death, meaning genetically programmed death, where the cell will die because it's gotten a signal to do so, not dying because it got chopped in half, okay, or got damaged by a virus or something like that, an actual signal, okay? And so you might want to guess then, one of the things that really has to happen to get cancer in you and in anybody you know who does get cancer, one of the most common mutations that there is P53, okay? Because again, this is such a powerful guardian to protect us from developing cancer that in order for cancer to develop, we usually have to lose it, Okay? And if you lose p53, then all of this protective mechanism is lost, and you proceed on, and that becomes one of the big problems that it has. And it's a really good example of that kind of sensing. Is homeostasis working? Is that cell functioning? If not, maybe we should kill that cell off. Okay? What I also want to talk a little bit about is yet a whole nother type of homeostasis. Okay? And this is one that gets a little bit less attention and is more the focus of what I'm studying, which is basically protein homeostasis, which goes by the fun name proteostasis. Okay? And that is that we think about how much protein is being made in a particular cell and what it ends up doing. Okay? And so, for example, what we will have is an mRNA going to the ribosome, and we'll make a little nascent peptide. We'll make a short little peptide that comes off the ribosome, and that will begin to fold up into its final version of a protein. Now, protein folding is a very complicated process with multiple steps, and a lot of stuff happens in that, and there's possibilities for things to go kind of wrong. Okay? And so one of the common things that happens with protein folding is they often have a lot of helpers. Okay? And one of the bad things that happens when proteins start to misfold or do something wrong is they tend to stick together and form aggregates. So we often call these chaperones because they tend to help keep those things from sticking together. Okay? And we pull them apart, and these chaperones are just helper proteins that help the protein fold properly and keep it from going into some kind of bad state. And you can get these various kinds of bad states of this folding where large numbers of proteins begin to stick together. And oftentimes, one example I always like to give for this, just to think about, is egg whites, right? So if you ever just take a look at an egg white, that actually is a really concentrated solution of egg albumin dissolved in water, which is just a protein. Tons of protein, very soluble. It's a little bit sticky, but it's obviously clear when you look through it, or mostly clear. However, then you throw that in a frying pan, and those proteins are going to denature because you've heated them up. They don't fall apart into lots of little amino acids, but they just lose their structure. And they switch to this big amorphous aggregate. And this big mess just falls out of solution, becomes this big white crashed out solution. You, of course, can't see through it anymore, and it becomes more solid. And that's, of course, what we normally eat. You could, of course, still eat it just fine. But I think we all know that if you let it cool down on your plate and let it sit out for a while, it doesn't just go back to being a nice soluble egg white again. It's kind of stuck. And that can happen inside your cells as well. We're generally trying to avoid this. We really don't want to gum up the whole inside of the cell with a bunch of egg whites, right? That's really not something that we want to have. And then they're hard to get rid of and tricky. And some of them are much more dangerous than even just that of an amorphous aggregate. Some of these form amyloid fibrils and oligomers, which are really, really tough and stable protein complexes that you can't even sometimes cut up at all if they get certain size. If they get too big, too many of them, you can't even get rid of them. Okay? So this then gets us into a whole nother family of diseases which are quite common, which are neurodegenerative diseases, which are the kind of the hallmark of most neurodegenerative diseases is a misregulation of proteostasis. Okay, and so something happens with these where there's something when it comes to these proteins. Maybe we don't degrade some of those proteins well enough and they build up too high. Or there's frequently mutations in them. You inherit a mutation in this version of a protein. It still is okay and it still works, but maybe it's a little bit more likely to aggregate on you. And over the course of your lifetime, it builds up into these bigger aggregates at a greater rate. You can also have environmental influences. Certain conditions, drugs, and things in the environment can make that happen. And of course, sometimes just bad luck with unfolding of these proteins into that, as well as also just errors that occur in transcription and translation that can lead to that. And this occurs in all sorts of aspects of your brain. So there's lots of different proteins that will form these aggregates in different parts of your brain and different types of neurons. Okay? And I could talk the whole time about different types of these neurodegenerative diseases, but of course some of the famous ones are like Alzheimer's disease, ALS, Parkinson's, Huntington's, and these are all quite common diseases that often a hallmark of them is this aggregation of excessive proteins, sometimes inside the cell, sometimes outside of the cell. But what ends up happening is that long-term kind of abnormality of that homeostasis. Something is wrong with that proteostasis, that cell is under prolonged stress for a long time, And we have that mechanism in our bodies because we don't want to get cancer, that if a cell is out of homeostasis for too long, we kill it off. Right? That's a very important thing for us to survive. And as I said, even sometimes just to survive long enough to get born. So we do that very intensely, but sometimes it can be not helpful. Right? In this case, we don't really want these neurons to die. We'd like them to live as long as possible, because especially we're not really going to regenerate these. If neurons die, we don't replace them. And so as you can lose a few, start losing a bunch, these symptoms take a long time to kick in. These can take decades to develop. But eventually you'll start running out of some of these neurons, and you can develop various diseases with different clinical prognoses based on this. But the mechanisms are kind of the same. Okay? And it's proven to be a rather difficult thing to kind of get at. And we've put a lot of money and a lot of research into studying these, and we haven't made an enormous amount of progress on these. The treatments today for Alzheimer's aren't that different than they were 30, 40 years ago. Okay? While cancer, for example, has changed quite a bit. Okay? So what I also want to do is focus a little bit more on what these proteins are doing inside the cell especially, okay, and where this stuff is going wrong. Okay, so inside of an animal cell, inside one of our cells, we'll have a bunch of different membrane-enclosed organelles. Okay, and one of the ones that I'm particularly interested in is the endoplasmic reticulum, which I'll just call ER for short. Okay? And that is going to surround the nucleus. It's often going to be covered in ribosomes. So a good chunk of proteins that a cell makes, on average about a third-ish, are going to get made there, and they're going to go dumped inside the ER. Okay, the ER has what we call lumen inside that, and a lot of those proteins are going to fold in that environment. And then they're going to traffic through what we call the secretory pathway, where they go out of the ER and the little bits bleb off and they go to the Golgi, and then those are then sent outside of the cell. And that would include any protein that needs to go to the surface of the cell or outside of the cell goes through that. So that would be hormones and surface receptors, extracellular matrix, a lot of things would all go through that kind of system. And the thing about the ER fundamentally is it has a finite capacity for handling proteins. There's only so much that it can do. A lot has to happen. The protein gets made, gets dumped right into the ER. We have to fold it, we have to process it, we have to shuttle it. There's a certain amount that can go through that without causing problems. So again, we have this proteostasis at the ER level, where we're detecting a certain amount of protein getting made, and we can't really exceed that. And if anything goes wrong, we're again getting out of our kind of homeostasis that we want to do. Okay, so I love signaling pathways, and I would love nothing more than to spend the next 15 minutes going through all of these pathways in glorious detail and talking about which site gets phosphorylated and where the protease cuts this site, but I will refrain. This is a kind of a cartoon that sort of shows a little bit about what's happening here. Okay, I will have to go through one of the pathways that that we're actually talking about. So here's an idea of ER stress, and we use the term ER stress for we're kind of out of homeostasis in the ER. We're out of that proteostasis. We have too much protein getting made, too much protein is trying to be secreted, and as a result, some of it is misfolding and accumulating in the ER, kind of gumming up the works. So we essentially have the egg whites building up inside the ER, preventing all those functions from happening. Okay? That alone is something that can kill the cell. You can block the release of necessary factors that it needs to release. It can just get blocked by that. Okay? So it has to try to fix that. So it'll activate 3 distinct signaling pathways, okay, which are themselves quite complex, which was what makes them fun. Together we call all 3 together, we call it the UPR, or unfolded protein response. Okay, so if we have ER stress, we then activate this response that we call the unfolded protein response. Okay, so the way this works is we activate a couple of things that have overlapping functions. So the way this box is drawn, so we have the 3 branches, which is simply named by the main signaling molecule in them, ATF6, PERK, and IRE1. Okay, PERK will be the only one we come back and talk a little bit more about later. Okay, but all 3 are kind of boxed here because one thing that they're going to do is activate some various transcription factors, and those transcription factors are going to change how much mRNAs get made and how many proteins get made that are especially involved in proteins that increase the folding capacity in the ER. So especially those chaperones I mentioned that help with protein folding. So again, let's think about this again as feedback. Everything's about feedback in all these pathways. And so we're making too many proteins, more than the ER can fold. So one of the ways to fix that is give it more helpers to help it fold more. So we can get rid of that problem and fix that. Okay, so we should be eliminating that ER stress. And these kind of arrows with a block on it, if you ever see that by a cell signaling nerd like me, that means inhibition. So it's going back to inhibit something. Okay? Now, these other two branches, PERK and IRE1, do that same thing, but they also have another side branch to that. And that is especially where they affect translation or the production of proteins. Okay, so PERK, for example, will cause the cell to make less protein total. Okay, it'll inhibit translation or protein synthesis, meaning we'll make less protein. IRE1 will generally destroy a bunch of mRNA. And if there aren't mRNAs around, they're not going to get translated and made into protein. So again, let's think about what that's going to do. It's going to be kind of the same idea. If I'm not making as much protein as I was before, guess what? I'm not going to be dumping as much protein into my ER. As a result, I'm not making that problem worse. So if something is wrong with the homeostasis, somehow I'm accidentally making too much protein, I'll just maybe back off a little bit, right? And so if you're burning your souffle, maybe turn the temperature down a little bit, right? I mean, just like kind of that kind of idea of trying to get back to homeostasis, right? Okay, so if I zoom in on this pathway, I am going to have to talk about PERK a little bit. Okay, and again, all these pathways are multifunctional. Okay, and so this stuff gets kind of fun. Okay, at least to me. Okay, but what we have here is PERK is a particular type of protein which is called a kinase. Okay, and a kinase is a protein that sticks phosphates on things. Okay, in this case, a protein. So this protein is also a fun one called eukaryotic initiation factor 2 alpha, or EIF2 alpha, right? Not that important what it is right now, but what it happens is if it gets phosphorylated, if you stick this extra little phosphate on it, that changes its shape, changes its behavior, and causes it now to inhibit any new protein synthesis, or what we call translation. So we're going to have less overall protein synthesis than we would have had before. So again, as the kinase, we just take this protein, modify it with that phosphate, and essentially turn that biological process down, and we inhibit it. Okay, so we make less protein. Okay, this pathway does other things too. So it'll activate some downstream targets that will happen, and one of those is another one just called ATF4 that signals toward a bunch of pro-survival targets, things that help the cell survive when it's being exposed to this stress and maybe fix some of that damage. And another thing that it does activate is something called autophagy, which is spelled autophagy but pronounced autophagy for some reason. And it basically does mean, like it says, self-eating. So the cell actually consumes some of itself for energy. And that would actually, in this case, include unfolded proteins. So if some of these proteins build up in the ER, they'll actually just get consumed, dragged away to the lysosome and chopped up into little bits. And that can help clear some of the excess proteins that happen. Okay? And so, again, what do I study? So I told you I really love feedback inhibition. So what I study is feedback inhibition of feedback inhibition. Right? Makes sense to me. Okay, so what I study are proteins that turn this pathway back off again. Okay, so the whole thing is this is a stress response pathway that comes in when we have stress to make less protein or to eat up some of it if it's unfolded. What I study are a couple of proteins with bizarre names, GAD34 and CREP. Okay? And so what GAD34 and CREP do is they send this pathway back. So what they do is they bind onto another protein. There's another type of protein which is a phosphatase, which simply does the opposite of what a kinase does. And that means it removes a phosphate from things. So if putting a phosphate on it decreased protein synthesis, taking the phosphate off will put the protein synthesis back on again. So this is to send the pathway back to where it was at the beginning. So especially if the stress has been resolved. Okay? We've actually had the stress, we fixed the problem, now we go back and turn things back to normal. Because we don't really want to keep translation shut off forever. We eventually need some proteins. Okay? So we do things like that. Okay? And so what ends up happening is it does also turn out that this protein that shuts this pathway off, especially here at GAD34 in this example, is turned on by that pathway. That's why it, again, is feedback inhibition. So as we have the stress response over time, my protein GAD34 will get turned on, and that will shut the pathway back off again. Okay, and that's a classic kind of feedback inhibition. So this is just an experiment I did a very long time ago, just to kind of show you what that kind of looks like. Okay, so what I'm doing here is I'm doing something called a Western blot, which is where I just use an antibody to detect a protein, how much it is. So basically this kind of darker spot here, the darker that band is and the bigger and thicker that black spot is, means the more of that particular protein is present. Okay, that's all it really means. And so what I'm doing is I'm testing these same cells with 3 different proteins. Okay, I'm testing against my protein of interest here, GAD34. That's the one that's the feedback inhibition of the pathway. Okay, and I'm testing against the phosphorylated EIF2 alpha. I can actually detect if that EIF2 alpha is phosphorylated. So if I go back to this previous slide, I actually can detect how much of this is here and detect how much of that is there. Okay, just by seeing how dark that little spot is on the gel. Okay, and then I have tubulin here, which is just always going to be there, just a control. Okay, and so I think what you can sort of see here is if I take a look at this response, is that at time 0 means that this cell has not been exposed to any stress yet. I then at that point treat it with a drug that causes some proteins to start to denature, and we start to cause ER stress. What you'll notice is EIF2 alpha phosphorylation shoots up right away. Right, so if I go from 0 hours to 1 hour, we see a much darker kind of band there. And that's just a sign that there's more of that phosphorylation. Okay? But as you'll notice, in an unstressed cell, we can't even detect the GAD34 there. Okay? We still can't detect it at 1 hour. At 2 hours, we can kind of barely see it. And then at 4, starting to build up. But what does GAD34 do but shuts this whole pathway back off again? So guess what we see is we see the phosphorylation go up, and then we see it go back down again. Okay, so it kind of have it in a cartoon here. If we see the phosphorylated EIF2 alpha, it goes up and then back down, and the GAD34 builds up over time. Kind of a classic of this kind of feedback control of this pathway. Okay, and that sort of sets up the kind of hypothesis that we have in a lot of our systems where the cell will respond very differently depending on if you're at the beginning of this process or at the end. Okay, at the beginning of this process, most protein synthesis is shut off. Toward the end, protein synthesis is back on. But if the stress is continued all the way through that process, and we haven't fixed the stress, and when I use one of these artificial drugs, you can't fix it. That's the whole point. It just messes with the stress too much. What you would then end up getting is we have our previous pathway that was all good and about survival, and we can fix this system, and we can fix the problem, and we can keep moving on. But now we have a system where I can't fix it, and the stress is continuing, and I've waited so long that GAD34 is built up. Okay, now I've got a lot of these proteins, and what will end up happening is we will activate eventually, more slowly out of this pathway, pro-death genes. Okay, and one of these is called CHOP. Kind of a fun name, just chops the cell up. Okay, and that's going to signal for apoptosis. So if that signal goes on for too long, we let the signaling events continue, it'll switch to an apoptosis mechanism. Kind of like that P53 example I showed a few minutes ago, where we can actually try to fix it, but there's a timer to it. We can only let that happen for so long. If the stress just keeps going, it's better to kill the cell for the benefit of the organism. Okay? And so what we've actually been able to do, and in a lot of other labs, is develop small molecules that inhibit these proteins. Okay? So these proteins are inhibiting this stress response pathway. So what they've actually been able to do is develop some small molecules that can block it. So for example, there was one invented now about 20 years ago called salubrinal. Okay? And it inhibits both of these proteins, GAD34 and CREP. Okay? And then there's a newer one that came out more like 10 or 12 years ago called cephin-1 that only inhibits GAD34. Okay? And they prevent those from being that inhibitor of the stress response pathway. So then what's going to happen in a patient if you do that? Well, the stress response pathway is not going to get inhibited. It's going to be on maybe longer than you would have expected it to be. So you're getting a stress response pathway that is stronger and more vigorous than you might have otherwise expected. So it's that same kind of idea of tweaking homeostasis a little bit. We have that feedback inhibition, maybe we could just tweak it and make it a little stronger and make that process work a little better. And now a cell that might have otherwise experienced a potentially lethal amount of stress, it should be able to take that level of stress and tolerate it. Okay? And these have worked really, really well in animal models, so in mice, and they've actually moved on into clinical trials for certain types of neurodegenerative diseases. So, for example, ALS, it works really well in, okay? And it seems to work better than even the top currently used treatments for ALS do right now, okay? And it seems to prolong the disease, makes it take a lot longer to progress, and weakens the severity of the symptoms, which is really quite nice. Doesn't cure it though, but that's often the case with a lot of these kind of treatments. Okay, and so this is kind of our hypothesis, right? So if we have this pathway, this is a pathway that can do both things. It can help fix the stress and help make the cell behave normally, and it can activate apoptosis. And a lot of that conversion from the early pro-survival to the late pro-apoptosis mechanism goes through these genes, GAD and CREP, okay, and these proteins. So if I can inhibit them, then what I should be doing is the part of the pathway that leads toward survival should be stronger, and the part of the pathway that leads toward apoptosis should be weaker. And that's just generally our idea. Okay? And again, this is shown here, again in a kind of cartoon form. This is kind of the same picture I showed a little earlier. So we might see the phosphorylated eif2 alpha go up and then back down again. We see GAD go up over time, and then over time as well, our pro-apoptotic kind of signaling goes up over time. So again, it's a time-based mechanism while that happens. Okay? And then our theory is if we can do things like inhibiting GAD34 and CREP, inhibiting our particular proteins, without GAD present, GAD doesn't go up because we've inhibited it. We will see the eif2 alpha shoot up and stay elevated longer. We stay in that early pro-survival mechanism longer, and so the later apoptosis mechanism doesn't happen as strongly as before. Okay? But there's actually a ton of unanswered questions about this, okay, from all aspects of how these drugs work. So, and it turns out with pharmacology, we do a lot of guessing, and we just test things and see if they work. And these drugs seem to work great. But we also don't really know how they work, right? And so there's a huge amount of controversy about them. You can get some pretty angry professors in a room debating how they function. And I mostly just kind of stand by and watch. But because I'm a little bit curious how they work, but I'm not really sure. It's also interesting that the drugs that inhibit GAD seem to be really effective at neurodegenerative diseases. The ones that inhibit CREP have a little bit more side effects and more problems we don't really want, and we really don't know why. Okay, so I've kind of mentioned GAD mostly, but if we look at them as molecules, there's very little, until I get into the really nitty-gritty about them, that's very different. There really isn't. They're very similar proteins. They both bind PP1 and encourage the dephosphorylation of eif2 alpha. There's not a lot about that. And when we've tested them, we've seen differences, some where GAD is really clearly induced by stress. The CREP original name for it was constitutive repressor of eif2 alpha phosphorylation because they thought it was always expressed, although that's clearly not the case now as we've gone more in depth into that. There's actually a lot of stress induction for CREP as well. Okay? And they have a huge amount of overlap in function in regulating this dephosphorylation, both in stressed and unstressed cells. And trying to tease apart what these two proteins are doing has not been very easy. Okay, we've actually had to do quite a bit of experiments to sort of see how GAD and CREP are different. Why do we even have two of them? And why do the drugs that hit one seem to work better than the other? And we really just don't know. Okay? And so what I've sort of set out is I'm going to briefly talk about why I've developed a zebrafish model to try to answer these kind of questions. Okay? So I think as you've seen from these kind of pathways that I've shown, everything comes down to timing in these pathways, right? The same pathway after 4 hours is somehow pro-survival, and after 24 hours is causing death. It's this kind of weird system, and every tissue that you study and every cell type that you study is going to have different kinds of time points where those things are occurring. And so you often don't really know where to start. Okay? And so this is where we turn to model organisms, where we can do a lot of these kind of experiments a little bit better. Okay? So if I were really trying to do this really hardcore, clinically relevant to really help medical treatment in patients, I'd probably do these experiments in mice, and other people are. But mice are expensive to make these transgenic mice and knockout mice, and you often don't have a very big sample size. You have to surgically cut into the brain to get your samples. It's not going to be you're not going to be able to cut into the brain of a mouse and look at 1 hour and 2 hours and 3 hours and 4 hours after stress and watch the pathway go up a little bit and down in the same mouse, right? You're going to kill the mouse by dissecting the brain out, right? So what I've actually done is I've chosen zebrafish as my main model organism because it gets us around some of these problems. Okay, so zebrafish are just a little tropical fish. You can buy it in any pet store. They call them Danios in the pet store usually. And really common, really easy to raise. I have thousands of them in my lab in just a whole wall of tanks. And what they do is I don't really do any experiments on the adults. I raise them and they basically swim around in warm water and I feed them every day and I change the water. And then what I do is I study the embryos. So they lay eggs about every week. And each female will lay sometimes 200 to 500 eggs at a time. And the eggs are all laid externally. Okay? And so they're really valuable for studying development because those early stages of development, gastrulation, blastula stages, and all these fun things are happening externally. Right? So imagine if you wanted to study these in a mouse, I'd have to dissect a mouse mother and look at the uterus to see the little implanting embryo. That's another pain to try to do. The fish are just laying the eggs on the outside, so no problem, right? The other thing about it is this is only a 24-hour-old embryo. So 1 day later, it's got a head and a tail and a digestive system and a brain and eyes, fully responsive UPR pathway. So the pathway that I study is fully functional, and I've got these, and these are absolutely tiny. So if you go to a sushi restaurant and look at Tobiko, it's about that size, okay? And because that's fish eggs also. And so I can put these in, I can have hundreds of them just in little dishes in my lab, do different drug treatments on them, collect them at 1 hour, 2 hour, 4 hour, 8 hour, and have all these experiments and see these pathways go up and down quite easily, okay? Another thing is there are also other model organisms that maybe are a little bit easier to work with out there in the world, like things like fruit flies or nematodes or things like that, but they suffer because they don't have both GAD and CREP in them. So zebrafish are kind of that middle ground where they're fairly simple, easy to work with, but they have both GAD and CREP. So I can kind of study what they're doing. Okay, so what I've done in my lab is I've focused on a few kind of key areas. So one thing I've done with these is I've focused a lot on apoptosis in the fin epidermis, meaning the skin around the fin, because that will actually behave a lot like the neurodegenerative diseases that I've talked about, where the cells are already under a lot of ER stress, and you can kick it over into apoptosis if you just tweak it a little too high. Okay? I've also done a lot of stuff with fatty liver disease, where they'll actually accumulate a lot of fat in their liver, develop steatosis, and develop things like cirrhosis. And these unfolded protein responses have a strong effect on that. I'm not going to have time to talk about that today, but that's been another area. And what's been interesting is the function of my genes is opposite in these cells. What I'll talk about is in number one, knocking out my genes is protective. It helps inhibiting them makes those cells less likely to die. But if I knock them out in the liver, it actually makes fatty liver disease worse. And so again, it becomes context dependent kind of thing. We're trying to figure out why is it helpful in some cells and not others. Okay, so a lot of this work has been done in mice. So we've done a lot of experiments with mice. And so these are baby mice that are born with knockouts in, this is the actual official gene name for CREP. And what ends up happening in these that we've kind of seen is that when you knock out CREP in mice, they have some pretty small growth. I think you can just tell this one's smaller than its litter mates than it should be. They also get pretty bad diabetes and anemia and will die at a fairly young age. But the GAD34 knockout mice, they actually develop just fine. So if I get rid of those genes from mice, you really don't see anything wrong with them. But if I knock out both genes, they don't even implant in the uterus. They don't develop into an embryo at all, and they die. Okay, so if you see a phenotype like that, that does tell you something about these genes, that there's some overlap in function. Because if I get rid of both of them, the embryo is just dead, right? I need those genes in there to get an embryo out. However, if I get rid of one or the other, I actually can survive, and that's actually also probably why sometimes the drugs that just hit one or the other are a little bit better able for us to tolerate than hitting both of them. And again, people are studying them in mice and trying to understand a little bit more about what's going on, but my sort of niche was just to look in fish and do the same thing. And I have mutants as well in my lab that are lacking each of these genes. Okay? And I'd also point out that there are human beings without CREP as well. It's fairly rare. There's only been a few, but there have been several babies born that have a mutated CREP. And just like we saw with the mice, they often have some growth defects. They're quite a bit smaller than they should be, delayed neural development, usually pretty bad liver cirrhosis. One of the things I mentioned I see in my fish as well, where I get too much liver damage, and also diabetes as well. Because it does also turn out that the cells that make insulin, that's a hormone that goes through the secretory pathway, those are under actually a lot of UPR stress as well. So there's a lot of interplay between these kind of diseases. Okay, so why focus in on the fin epidermis? So this is just from an older paper, actually from a friend of mine, that he had done many years ago, where he had looked at 24-hour zebrafish embryos and had looked at them and tried to monitor apoptosis in these embryos. So what he's doing here is using a stain, and the stain will make these little fluorescent white dots every time a cell is dying. Okay, it's technically something called tunnel staining. I don't want to go into all that, but it's just a way to see a cell that is undergoing apoptosis. Okay? And if you take a look at, this is a control embryo, and an embryo that means no stress has been added. There are a few cells dying. As I've said, it's pretty normal for cells to die at times during development. It's a normal kind of process. However, if I treat with a drug, and this drug is just called thapsigargan, that promotes unfolded proteins, I cause that, you'll notice that I cause a ton of death, but not throughout the entire embryo. Right? You notice the whole embryo doesn't turn white. The whole embryo just dies. I'm not giving it that much of the drug. You actually could if you put enough in there, but I'm just putting a little bit in there. I'm tweaking it up to the particular cells that are most sensitive, the ones that are already under a lot of ER stress. Okay? So for example, I mentioned it's the tip of the tail. The tip of the tail is already under a lot of ER stress signaling to begin with because it is rapidly growing at that stage and is growing all that cartilage, all that extracellular matrix, and that is all proteins that are going through that secretory pathway and going out of those cells as they're growing. So their ERs are just jam full of laminin and collagen. Okay? And so if you tweak them with a little bit of this extra drug, they're already at the edge of homeostasis. They go right out of it, and the cells start dying. Okay? And so you can see that quite clearly. They also get it in the eye and in the brain and a few other areas, but the tail is really obvious. Another thing about the tail that's really nice is if you're lazy like me and you just put them down on a microscope, the tail just lays flat and you can just look at it pretty easily. And so it makes it a pretty easy thing to look under a microscope. Okay? And what they've been able to do in these guys is, hit them with stress and look at what's happening with that apoptosis in these cells and look at different time points. And what they were able to notice that if they looked early on and they just whack this embryo with a ton of stress, they will be able to get early induced apoptosis. Okay, that happens in this case 4 hours after you treat with stress. And that goes through a gene that is actually very similar to the P53 I talked about. Okay, and it's cleverly called P63. Okay, but it'll go through that and another little helper protein called PUMA. Okay, and we sometimes call that the acute cell death. It happens very rapidly. The cell is already on the edge of homeostasis. You whack it with this stress and it goes right into apoptosis. Or it can have a kind of a prolonged stress that in this case will go out over 24 hours. and you've treated it with that stress for a long, long time, and then that'll build up, and it'll go through a CHOP-dependent death where the cell will die through that. So we actually have different genetic ways that the same cells can die depending on timing. Okay? And so what we kind of set out in my lab, this is really where I started working on this project, was what role do GAD and CREP have in these processes? Okay? Is GAD and CREP, are they involved in this CHOP side? Are they involved in this PUMA type of death, or both? Right? Basically have no idea. And again, with fish, it's a very good way to do this. We can test the different time points, see the different stages, see how these pathways change, losing these individual genes. Okay? And so one place where we might want to start, and this was work done by one grad student in my lab, Krithika, and she did a lot of work that I've summarized into one cartoon, which I feel bad about, actually, how much work it was. But she went through and looked at the expression of all of these genes in excruciating detail at excruciating large number of time points, just to see how they go. Okay? And what she saw was if you start treating with stress at 24 hours, so the timing gets really confusing here. We grow the embryo up for 24 hours, and after 24, we just call that time 0. Okay? Then that's when we whap it with stress, and then we wait. So then 4 hours after that, we look and see, is there apoptosis or is there gene expression changes? And then out to 24, we look. And we actually go past that. We actually went as far as 48 hours just to see how gene expression changes after that stress. Okay? And so, for example, we might have here a positive control. This is one of the chaperones, one of the downstream genes that we know goes up when you have unfolded protein response, when you have ER stress. And so what we see is it goes up real fast, stays up for a long time, and after long enough time period, it starts to drop back off again. Okay? We also have here our GAD34 and our CREP, and we see that they both go up, with CREP actually going up a lot, and then after time dropping off. GAD will typically settle back to where it was before. CREP will actually go below where it was before and actually drop to lower levels than what it would have had initially. Okay? And then our pro-apoptotic gene CHOP really doesn't go up that much early on, but later it's really starting to build up over time, and we build that high levels of CHOP after a long time period. Okay? And so we've kind of done a lot of work with that, and we're seeing that our genes are changing quite a bit over time. And so that again is the strength of these as inhibitors. There's very little inhibition at first when the pathway starts. After a few hours, now these inhibitors have really built up and they're blocking this pathway very strongly. But after a while, they start to go back down again themselves. So there's a lot to look at these individual stages of what's going on. Okay? And so in the next thing is actually stuff that Dr. Hicks did, or Professor Hicks did when she was in my lab of a lot of her master's thesis, where we could actually go and see what happens physiologically if we eliminate the function of these genes. Okay? And fish, again, have several other advantages that we can do when we're playing around with things, and that is that we can inject at the one cell stage, right when the egg is laid. We can inject into that one cell, and every cell that is going to come in that total embryo comes from that one cell. So if I fill that up with a drug or fill it up with mRNA or any kind of reagent, every cell in that fish is going to get that. Fairly easy to do. I can raise them up and then just look at the tip of the tail and stain with a dye that will stain dying cells bright green. So I can see what's happening if I eliminate the function of my genes or test them out in different ways. So again, this is our kind of hypothesis that I had showed before, and we have our whole pathway. So why don't I start with salubrinol here, okay? Just a drug, one of these drugs that we're testing on humans as a protective neurodegenerative drug treatment, and it inhibits GAD and CREP. What if I treat with that? Well, our hypothesis is it's inhibiting the inhibitors of this pathway, so the pro-survival part of the pathway should be on a little bit more than you would have thought. So we should get more of this pro-survival signaling, so maybe these cells will survive better than we would have thought. Okay? And so exactly what we do is collect the embryos at fertilization, keep them for 24 hours. After 24 hours, give them a little bit of the salubrinal for 2 hours, and then whack them with stress. Okay, after you whack them with stress, you then look at 4 hours, and you look at 24 hours. Okay, you just take those embryos, you stain them with that dye, and the more green cells means the more dying cells. Okay, and so what we take a look here is we again just zoom in on this picture, right? We just mount the embryo, just zoom in on this little area of the fish, and take some pictures. Okay, and what we'll see is if we have control, meaning no stress, very little death, treat with salubrinal, very little death, right? But that's what's to be expected. There's not much death going on with no stress being added. Okay? However, if I then do whack it with stress, I then give it the drug that induces that, I think it's pretty obvious that this looks a lot more green than that. Okay? And that is cells that are dying in there. Every green cell is one that is going into apoptosis early. Okay? And so as we see that, we see the tip of the tail is very, very sensitive to this. We see massive apoptosis in that, just with a fairly subtle induction of stress. However, if we pre-treated with salubrinal, what we'll see is if we look at 4 hours, or if we look at 24 hours, we see less apoptosis occurring in those time periods. So we've treated with this inhibitor, they're able to take the same amount of stress, but a lot fewer cells are dying. Okay, so it does seem to kind of fit with what we kind of hypothesized for what they're doing. Okay, and so when Danielle was in my lab, we also designed some other little tricks that we did, okay, where we could block the function of these individual genes, just specifically GAD and specifically CREP. So what turns out is that when a gene gets made, what we end up having is in the mRNA, we have introns and exons, okay, in a zebrafish. And this is, what that means then is that as this comes off the DNA and is read as a pre-mRNA, has a bunch of RNA in there that's going to have to get removed and then spliced back together again. Okay, to make the final mRNA that will encode for a protein that actually works. So what we actually can just do is simply design a little reagent that will bind between the exon and the intron called a morpholino. That will base pair there and prevent that splicing from occurring. And so you end up retaining that intron when you don't want to. Okay, so you now have essentially junk mRNA in the middle of your mRNA. Okay, and so it's not going to make a protein that does anything. Okay, and so now we basically are able to block the function of these proteins whenever we want to. Okay, and that's basically one of those things. We've actually come back and done this also with mutants. I mentioned I have the mutants. I'm just showing you, I'm not going to show you all that data. I'm just showing you the ones with the morpholinos. Okay, so this again is our hypothesis, right? We showed that the drug that inhibited both of them would give us more survival. Maybe I can knock out GAD or CREP or both, because I can just mix the two morpholinos together and knock out both things. And then I can just see what happens. Okay, so we do the same kind of experiment, collect them, except I inject right at the one cell stage. We inject it right in there and put that morpholino in there, grow them up, and at 24 hours treat them with stress and then measure the amount of apoptosis 4 hours later or 24 hours later. Okay? And so what Danielle saw when she did this experiment was that we would see here with in the wild type or the morpholino injected ones, the ones lacking GAD34, what we saw if we looked really quickly, that rapidly induced death, there was a slight decrease, but it wasn't statistically significant. But if we looked at that later 24-hour death, the one that happens from just prolonged death, again, more like what we would probably expect in neurodegenerative disease, we saw a drop-off. Okay? So if you eliminate GAD, we see quite a bit less death than you would have expected. So again, we can kind of make that cell more resistant to the same amount of stress that would have caused more damage before, can now tolerate it a little bit better. Okay? What we then saw, if we knocked out CREP, our other gene. Okay, again, they biochemically look really similar. They both get induced by stress. They're really hard to tell apart. But when I knock out CREP, we really don't see anything. Okay, we've seen this both when Danielle did this with morpholinos, when we've tested it with the mutants. They really don't seem very different. So eliminating CREP doesn't seem to do much. Okay? However, when we eliminate GAD and CREP, get rid of both of them, we actually see a stronger effect. We see protection at the 4-hour time point, that acute death is actually less, and the death that comes from the prolonged chronic stress is also less, and it's less by a greater amount than even just by GAD by itself. So what that tells us is we can kind of conclude a few things from these experiments, right? That when we look at apoptosis, that when we can eliminate GAD, we really protect against that long-term death, but not so much the quick, rapid onset death. We knock out CREP, it doesn't really do much, but we knock out both, protects against both. But also when we use a drug that inhibits both, it protects against both. Okay? And so it's telling us that, again, that the same amount of stress over different kind of time periods is resulting in a slightly different kind of genetic outcome, and that really our two genes are behaving a little bit differently. Okay? And so these are essentially kind of the conclusions that we can kind of make from this, is that GAD and CREP are both induced. They're both stress-induced genes, and I'm doing a lot of more experiments to study that right now, of looking at their promoters, looking at their translational regulation, looking at how they get degraded, just understanding how they go up and down with stress. Okay? We're also we see that they seem to be partially redundant, that if I knock out both, I get a much stronger effect than if I knock out one or the other. Okay? And again, we're still trying to dissect that out. How much do they seem to be redundant in other functions of that? So I'm doing the same kind of stuff in liver, trying to piece that out as well. And again, especially when you knock out both, or in the case of chronic, just GAD, you get protection against this long-term stress-induced apoptosis. So again, it seems to be a decent model of how these genes are working. And this kind of hypothesis, at least in the fin, seems to be true. That if I eliminate the function of these genes, I get more of the pro-survival signaling, less of the pro-apoptotic signaling. So that whole idea of taking the homeostatic signaling, just tweaking it a little bit, and tilting the balance towards survival seems to work. And if we can kind of mimic that in neurodegenerative diseases, we should have some better ability to treat those kind of treatments. Okay? And these are just, it's obviously not a clinically relevant system. I'm not trying to cure, fin cell death in zebrafish as the great disease of the future, but it is something that we can make some advances on and do some learning with, okay? And learn a little bit more about these pathways. Okay, so I just want to end there and do a couple of quick acknowledgments. So another professor in my department, Lisa Rishnick, we've done a lot of work together. She's actually a lot better neurobiologist than I am. I'm very much a cell biologist, biochemist. She's more of a neurobiologist. We've been doing more neuronal stuff with her, and we've also been doing a lot of gene expression stuff. These are some of the various graduate students who have worked on this project over the years in my lab. Everybody has their own little piece of the puzzle that they've worked on, and I showed you a little bit of Danielle's work and Krithika's work in that, but we have other students have done other pieces of that puzzle. Okay, and I have 3 graduate students currently in my lab, and I want to give a quick little plug out for our program. I know many of you are undergraduates here, and our master's program is actually a pretty good place if you want to get more experience in research, you want to do research of some kind. You do need to get a bachelor's before you can apply for ours. Associate doesn't quite work, but if you get a bachelor's and then maybe down the road think about us, a couple of big advantages. One that's very big is we offer a teaching assistantship, where if you teach our labs, your tuition is free. So the price is right on that one. And we pay a kind of embarrassingly small stipend also that you can also use to pay for housing and maybe some food. But at least the tuition's free. And it lasts about a 2-year program. You can get a lot of teaching experience, a lot of research experience. And honestly, the person the program is best for is someone who doesn't know what they want to do. And you want to explore, try something. Like, I'm not sure if I want to do biochemistry or ecology or what. You can just try those things out before you just jump into some PhD program or something that's going to eat your life away. Okay? So just want to point that out. And I also want to talk and thank some of the funding agencies that have funded this over the years. And I've had grants from the National Institutes of Health and the National Science Foundation as well that have funded this. And I do want to just give a quick plug out for how important scientific funding is for doing this. I really couldn't do any of this work without these kind of grants in the past, and I think a lot of people who maybe are undergraduates sometimes don't know how expensive and time-consuming these experiments can be, and the training of graduate students and the training of people requires this kind of funding. And I think you guys may know from the news, there's been a lot of kind of cutbacks and some pretty sad kind of things that are happening with scientific funding. And I just would encourage people to pay attention to that and just kind of know how important these things have been over history. And I always think back, so my PhD work was paid for by fellowships. I had some from the NIH and the Department of Defense, paid for my PhD. And I remember my dad got the very first year in 1950 when they did NSF fellowships for graduate school. He got one of the first ones. And that was allowed him to get a PhD and do a lot of stuff. And unfortunately, a lot of these are getting frozen right now. And a lot of people are very uncertain about where they're going in their careers. And I just think it's something to take home, and just with a lot of news out there, but if you ever let a congressman know just how important NSF and NIH funding are to do this. I was able to take a lot of students to conferences, took Danielle to a conference, and you get a lot of kind of networking, paying people over the summer, buying equipment, things that just never would have been possible without these kind of agencies. So that is what I've got. So.